Hello students. So today we shall study the anomalous secondary growth in Dracaena. In the previous video, we studied the anomalous secondary growth in Bharavia. Dracaena is a monocot plant and you can see on either side of the slide the indoor Dracaena plant. So this is Rekha presenting you from Government Science College, Bangalore. So what is this anomalous secondary growth? So in many plants, the pattern of secondary thickening or secondary growth shows deviation from the normal type. So this deviation is called as anomalous secondary growth. So it is more common in tropical plants. So this anomalous secondary thickening is not a disease or an anomaly, but rather it is an adaptation to suit the habit and habitat of the plant. In dicots, vascular bundles are conjoined, collateral, open and arranged in a ring. So open means the cambium is present and it is open for secondary growth. Formation of secondary tissues takes place by the fascicular cambium in the stellar region and the cork cambium in the cortex region. In monocots, vascular bundles are conjoined, collateral, closed, and they are scattered in the ground tissue. So we do not see the secondary growth in monocots. But in some plants, secondary growth is seen, which is not the normal type. It is deviation from the normal type, which is called as anomalous secondary growth. So why is this anomaly? See, anomalous, it may be due to the primary structures or it may be due to the secondary structures. So what are the primary structures responsible for anomaly? So firstly, occurrence of scattered vascular bundles in dicotyledons, arrangement of vascular bundles in a ring in monocotyledons, occurrence of medullary or cortical vascular bundles, occurrence of intraxillary phloem, occurrence of separate phloem bundles. The anomalous secondary structures, it may be due to the abnormal behavior of normal fascicular cambium, abnormal behavior of abnormal cambium, replacement of normal cambium by abnormal accessory cambia, or the origin of cambium in monocots. So this is the flow chart representing the primary, secondary and anomalous secondary growth. The primary growth in plants, it is called as the increase in the height of the plant, which is due to the apical meristem. The secondary growth in plant, it is due to the intercalary meristem, which increases the girth of the plant. Whereas the anomalous secondary growth, it may be adaptive or non-adaptive. So adaptive anomalous secondary growth is to cope up with the external condition, whereas the non-adaptive, it is due to the variation in the cambial activity. So now let us study the anomalous secondary growth in Dracaena stem, that is the monocot plant. So the secondary thickening is usually absent in monocot plants since the vascular bundles, they are closed type and they are scattered. Closed in the sense there is no cambium. So however, in few plants of monocots, they show the anomalous secondary growth such as Dracaena, Yucca, Aloe, Sansevieria and Agave. The secondary growth in monocots occurs by the abnormal growth from the abnormal cambium. So in monocots, the vascular bundles are closed and the cambium being absent, the secondary growth is absent. But in some plants like Dracaena and Yucca, the secondary growth takes place. So the normal cambium is absent. The abnormal cambium may either develop from the cortex or pericycle and show abnormal activity. So secondary growth is due to the extrastellar cambial ring in the cortex or the abnormal activity of the cambium. So during secondary thickening, what happens is the young stem has an epidermis followed by sclerenchymatous hypodermis, a large number of closed collateral vascular bundles. They are scattered in the ground tissue. So one of the outer layers of cells from the ground tissue becomes meristematic and functions as cambium. So here the meristem will be named as secondary thickening meristem. The cambial cells are fusiform or rectangular in shape in alternate patches. The inner parenchyma cells are called as the conjunctive tissue. The cambium, you can see the diagram here, which represents the different stages of secondary growth in Dracaena. So the cambium, the black color line represents the cambium, the blue one is parenchyma, and the red is the xylem. So the cambium, instead of forming phloem and xylem on the outer and inner sides as in normal condition, the cambial ring produces xylem and parenchyma to the inner side first and later small amount of parenchyma on the outer side. So the amount of parenchyma produced outer side is very less. So after a while, the activity of the cambium on the inner side changes, and above the xylem, it starts forming phloem and then again a xylem. So thus the phloem gets encircled by a ring of xylem. So such a type of vascular bundle is called as leptocentric or amphivasal vascular bundles. The xylem formed earlier has bigger vessels. And of course, around each vascular bundle, sclerenchymatous sheath is developed. So the cambium, after some time, it alters its activity and forms a xylem on the inner side at those places where it was previously forming the parenchyma and parenchyma in place of xylem. So after some time, the cambium again behave normally to produce another ring of vascular bundles. So the activity of the cambium, it keeps on changing regularly and more rings of vascular bundles are formed. So the last ring of vascular bundles is embedded in a mass of lignified conjunctive tissue. So in this diagram, you can see the secondary tissue, that is the conjunctive tissue, the cambium, and in this you can see a number of vascular bundles which are embedded. So cork cambium is also formed below the hypodermis, which forms the cork and the cork cambium in a regular fashion. So this is also the whole view of the transverse section of stem of Dracaena, showing the abnormal secondary growth. So to summarize, in most of the dicot stems, the normal structures with a ring of collateral vascular bundles are present. This ring cuts off secondary xylem on the inner side and secondary phloem towards outer side. However, a number of plants show secondary growth that deviates from the normal secondary growth. So this deviation of secondary growth or secondary thickening is called abnormal or anomalous. Thank you.